Okay, so assalamu alaikum everyone. My name is Maryam Altayeb, and so today we're going to be giving you past sessions for MOL, inshallah all goes well. Just before we begin, I want you guys to know if you have any questions, you can feel free to ask in the chat. You can feel free to stop me if need be, and I'm gonna keep repeating the points multiple times, inshallah, to ensure that you guys have everything down by the end of it. And also I'm working on two monitors, so if I'm looking away from the camera, I'm still looking at the screen so I can still see you guys and see your comments. It's just much easier this way. Okay, so let me just share my screen. Okay, so we're gonna start first by talking about the urea cycle. So and let me just open the chat on the side in case anyone has any questions. Okay, so bismillah. So first of all, we're going to start talking about the urea cycle. In order to talk about the urea cycle, we need to understand what is urea and why are we learning about this in the first place. So urea cycle is the way your body excretes ammonia. And we know that we have amino acids in the body, right, that make up proteins. We have fats and we have sugars. Sugars are stored as glycogen, right? And then we learned about that in very extensive, excruciating detail in MOL. Okay, and then we also learned about storage of fats, which is related to insulin and whatnot, and you guys are going to take that in a later PAL. But the problem with degrading amino acids is that it turns into ammonia, okay, and nitrogenous waste products. And there's no way to store these products. So there's not like, it's the way you store glucose is glycogen, right? And the way you store lipids is in fats. So there's no specific way to store these nitrogenous waste products. And the problem is if ammonia builds up in the blood, that's going to cause a lot of problems. And that can cause specifically CNS symptoms, so problems with the brain. And so because of that, the body has to create a way in order to excrete these safely, because ammonia is not safe to be excreted. You want to put it in another form. And so hence we have the urea cycle. Okay, and the urea cycle takes place in the liver, which we know is the detoxifier of the body. And it takes place in two locations. So within hepatocytes or your liver cells, few steps take place in the mitochondria, and then everything else takes place in the cytoplasm or cytosol. Okay, so again, what is the urea cycle? It's the way the body excretes ammonia in a safe way by turning it into urea. And we're going to learn about all of these different steps today. So first, I want you guys to take out a pen and paper or anything in front of you, even if you're going to write it on your phone, because I'm going to teach you two mnemonics that inshallah are going to help you remember all of the steps even before we start. And I'm going to keep reiterating these throughout the whole talk. So first we have the intermediates of the urea cycle. And so when we think of urea, we think of urine, right? Because that's excreted in our pee. And so I want you guys to picture this. Any person urinating in public will be prosecuted. Okay, and so under that premise, our first mnemonic is going to be ordinary careless crappers are also furious about public urination. So just picture someone really angry about people urinating in public, which is disgusting, first of all, but that's our first mnemonic. And hopefully the part about urination is going to help you remember that this is related to the urea cycle. Okay, so ordinary is going to start with ornithine. Okay, so that's, and all of these are in order. So just remember it with me. Ornithine, and then careless is for carbamyl phosphate. Okay, crappers is for citrulline. R is for aspartate. Okay. Also is for argininosuccinate. Furious is for fumarate. Fumarate. About is for arginine. Public is just a filler. And urination is for urea, obviously. Okay, so again, ordinary careless crappers are also furious about public urination. I have it written here on a sticky note, just keep in mind. So I would highly recommend you guys do it as well with me. So ordinary for ornithine, careless for carbamyl phosphate, crappers for citrulline, R for aspartate, also for argininosuccinate, furious for fumarate, about for arginine, and urination for urea. Okay, so just keep that in mind. That's our first mnemonic. Okay, and once you guys have that down, we're going to move on to the second mnemonic, which is how do you remember the enzymes related to the urea cycle. Okay, and so my mnemonic for this one is cola. So I put this very delicious looking bottle of Coca-Cola that is not at all tempting. But yeah, so cola. So just remember COLAAA, but the L is not included in the mnemonic. I just used it, to make it easier for you guys to remember. So the C is for carbonyl phosphate synthetase. The O is for ornithine transcarbamylase. Okay, well, the spelling is wrong, but I'll fix it. After, A is for argininosuccinate synthase, and this is where it can get a bit confusing. So argininosuccinate synthase, which means you're creating it. So first we need to make argininosuccinate. Okay, then we need to break it down. So argininosuccinase, and then our last enzyme is going to be arginase. Okay, so for this one, it's going to be COLA. So carbonyl phosphate, ornithine transcarbamylase, argininosuccinate synthase, argininosuccinate, and arginase. Okay, so those are our two mnemonics. So first we have ordinary careless crappers are also curious about public urination. And secondly, we have COLA in 3 days. Okay, and I know I spent a bit of time on this, but I really want you guys to remember this because it's going to make your memorization of urea cycle 100 times easier. Okay, so now let's take a look at the urea cycle. So in the urea cycle, we have NH4, right, or our ammonia, combines with CO2, aspartate, and 3 ATP and 2 waters to give us urea, fumarate, 2 ADP, 2 phosphate, 1 AMP, and okay. So I know this kind of looks confusing now, and we're going to go through it step by step. But what I just wanted to emphasize is that these are all of the different intermediates we talked about, right? So first, ordinary, if we're going for the first mnemonic, ordinary careless crappers are also curious about public urination. So ornithine, careless for carbonyl phosphate, crappers for citrulline, which transfers from the mitochondria into the cytoplasm. Okay. R is for aspartate, which is here. Also is for argininosuccinate. And then we have furious for fumarate, about for arginine, and then public urination for urea. Okay, so that's just how you remember all of the different intermediates in the cycle, which is going to help you guys answering questions. Okay, and then for the enzymes, again, COLA, so carbonyl phosphate synthase 1, and then ornithine transcarbamylase, which is here. And then you're going to have your other enzymes, which are going to be here, like argininosuccinate synthase, which is going to create argininosuccinate, and then argininosuccinase or argininosuccinate lyase, which is going to break it down into arginine and fumarate. And then we have arginase, which is going to give us our urea. So this is again the endpoint, and then our ornithine starts the cycle back up again. Okay, and I know I went through that quickly. We're going to go through all of the steps and details. I just wanted you guys to hear it in the beginning. Okay, so now we have the first step, which is, remember, carbonyl phosphate synthetase. That's the first enzyme and the COLA mnemonic. And so what carbonyl phosphate synthase does, and remember, we're talking about 1. So carbonyl phosphate synthase 1. Okay, so what it does is it's going to catalyze the formation of carbonyl phosphate from 1 molecule of nitrogen. Okay, and then we have our bicarbonate and we have our 2 ATP. And important for you guys to keep in mind is that this first step happens in the mitochondria. So if we look at the image here, this first step is happening in the mitochondria. But what's more important than that is that I really need you to remember that we have a molecule called N-acetylglutamate synthase, okay? And this is going to be creating our N-acetylglutamate. And why do we care about N-acetylglutamate? Well, N-acetylglutamate is what's going to catalyze this step. It's an obligate activator. So this has to be present. You have to have N-acetylglutamate in order for your CPS1 or carbonyl phosphate synthase 1 enzyme to work. Without this, it's not going to work and the whole urea cycle is not going to happen. And remember, that's bad because you're going to have a lot of nitrogenous waste products building up in the blood, which is very bad for you. Okay, so just know that N-acetylglutamate is an obligate activator that is present after a protein-rich meal. So that's when we eat proteins. And subhanAllah, the body knows that, let's say you're not just going to break down protein if it's not being consumed. So the more protein you consume, the faster this process is going to go because you're going to have more N-acetylglutamate. Okay, so this is the critical step in removing the ammonia from the blood, and it's irreversible. Why is it irreversible? Because you're breaking down 2 ATP to give you 2 ADP, right? So we said we need 2 ATP for the reaction. So this is where we're cleaving our ATP. And what are we making? We're making carbonyl phosphate. Makes sense, right? The name of the enzyme is carbonyl phosphate synthetase. So what you're doing is you're creating carbonyl phosphate, okay? And so this is the first step. And again, I just really need you to remember that N-acetylglutamate is very important. Without it, this whole process will not run. Do you guys have any questions? All good? Okay. I'll keep repeating the steps as we go along, but so again, if you guys have anything, just stop me and I'll re-explain. So that's the first step. We need carbonyl phosphate. So in our two mnemonics, so ordinary careless, we'll get to ornithine in a second. Careless is for carbonyl phosphate, which we just created by the enzyme in the colonmnemonic, which is carbonyl phosphate synthase. Okay, so that's the first step. Second step is ornithine transcarbamylase. Okay, so ornithine transcarbamylase, what are we doing? We're taking this carbonyl phosphate, okay, and we're turning it into, with ornithine, and we're turning it into citrulline. Okay, so what this is doing essentially is it's catalyzing the formation of citrulline. And here you don't have any specific activators you need to know about. The most important was N-acetyl, which was N-acetylglutamate that we talked about in the last slide. So for the rest of the Urea cycle, there isn't anything specific. So ornithine transcarbamylase is going to catalyze the formation of citrulline and one inorganic phosphate. Okay, we said again from ornithine and carbonyl phosphate, and this occurs in the mitochondrial matrix. So the first two steps, which are with the first two letters in the colonmnemonic, so carbonyl phosphate synthase and then ornithine transcarbamylase, these two steps happen in the mitochondria in liver cells. Everything else is going to happen in the cytoplasm. Okay, and so what happens is it catalyzes the introduction of one carbon atom and one nitrogen atom. So remember, the whole idea of the urea cycle is you're trying to excrete these ammonia groups. So you'll notice as we go through all of these steps, what you're doing is you're adding more ammonia, more ammonia. So you see how there was, here you can count 1, here you can count 1, 2. And so what you did is you transferred this here, right? So now you can count 1, 2, 3. So urea is basically loaded with all of these nitrogens, so we can excrete them, okay? And so now that we created citrulline, it moves into the cytosol. Okay, so now we're working with the third step in the cytoplasm. Do you guys have any questions about this step? No question is a bad question. No question is a stupid question. So genuinely, if you guys want me to repeat it, I will. That's fine. Okay, silence is also an answer. So I hope that was clear. And again, just to go over it one more time. So we have in the mitochondria, the first two steps. So you have carbonyl phosphate synthase 1, okay, takes all of these products and turns it into carbonyl phosphate, okay? And then you have ornithine that comes and combines with this carbonyl phosphate we just made by the ornithine transcarbamylase enzyme, and that makes citrulline. And now citrulline is going to transfer out into the cytoplasm where we can continue the rest of the urea cycle. And again, remember, for carbonyl phosphate synthase 1, N-acetylglutamate is an obligate activator, meaning if it's not present, the reaction will not happen. So it is very important, and levels of this are going to be stimulated by eating dietary protein. So think of like, eating chicken, meat, even, non-meat sources of protein. Okay, so that's step 1 and step 2. Okay, now in our other mnemonic, we move on to, in the first mnemonic, we said ordinary, careless, crappers, R. Okay, so crappers was for citrulline. Okay, and we finished the other two enzymes in the colon-mnemonic, right? We took C and O, and then we have our 3 A's. So first we need to make argininosuccinate. Okay, so the way you make an enzyme or you make a molecule is by its enzyme. So argininosuccinate synthase or synthetase. Okay. And just so you guys keep this in mind as a general principle in MOL, if it's called a synthetase, not a synthase, there's an extra T, that means it's using an ATP. Okay, so the first step used two ATPs, now we're going to use a third one. Okay, so ATP is going to be broken down to give you AMP and then the two phosphorus. Okay, so now we're taking our citrulline with our aspartate and creating argininosuccinate by the enzyme argininosuccinate synthetase. Okay, so in other words, argininosuccinate synthetase catalyzes the condensation of citrulline and aspartate to form argininosuccinate. So now they're one molecule. And so when we do that, let's count how many nitrogens. So in the previous steps, we added 3, right? It was 1, 2, 3. Now, how many more are we adding? We're adding 1 more, which you can see here. Okay. So far, this is loaded with 4 nitrogens. And we're just going to keep adding until it can be excreted as urea. Okay, so again, so third step is citrulline and aspartate, so crackers are, and then by the enzyme argininosuccinate synthetase, which is the first A in the colon mnemonic, is going to give us argininosuccinate, which is R also. Okay, so ordinary careless crackers are also curious about public urination. Okay, so again, here we're forming argininosuccinate. Again, if you guys have any questions, please let me know in the chat. And so that's the heaviest part of it. The rest of it is relatively easy. So now we have this argininosuccinate, but what we're doing is we're going to break it down. So when you break down something, instead of calling it a synthase, you can just say lyase or just use name ending in ase. So argininosuccinase. And that's going to break this molecule down into arginine and into fumarate. Okay, and then fumarate can be used to create glucose and add into the TCA cycle, and you guys are going to see about that in a second. Okay, so what they're saying here is this completes the transfer of the amino group from aspartate to make arginine. Okay, and it's going to retain the skeleton backbone of aspartate as a fumarate molecule, which is good. So you don't really have to worry about that, but what I want you guys to know is now we have arginine and we have fumarate. And the important thing about fumarate is that it's going to be used in other processes in the body. So, you don't want to waste anything. So that's why you can see that all of these biochemical pathways are very interrelated. Okay, so that's where we are so far. So argininosuccinase. So again, in the enzymes, we have CO, ignore the L, AAA. So now we finished carbonyl phosphate synthetase 1. We have ornithine transcarbamylase, right? And then the first A was argininosuccinate synthetase, which we finished from. Now we have argininosuccinase, and now we have our last enzyme to worry about. Okay, and for the molecules, we said ordinary careless crackers are also furious about public urination. So we said ornithine, right? And carbonyl phosphate, we finished from those in the first step. And then crackers is for citrulline, right, which translocated into the cytoplasm. R is for aspartate, okay, which we used to combine with citrulline to create argininosuccinate. And now argininosuccinate in this step, furious, is broken down into also furious. So fumarate, and then, furious about. So fumarate, and then we have arginine, right? So that's the step we're at. So right now we basically have one final step to complete the whole urea cycle, which is creating urea, right? That's the most important thing about this. So we have our arginine that we're going to break down one last time using a water molecule, okay, which is what we call hydrolysis, right? So by the enzyme arginine. So arginine is broken down by arginase, okay? That's going to give us ornithine, which remember was one of the first things we talked about in the urea cycle. So this is going to go back to the first step and just keep going on and on and on again. Okay, and of course we have our end product, which is urea. And you can see that this urea has 2 nitrogens, right? 2 ammonium molecules. And so this ornithine is going to cycle back into the first step, pick up another carbonyl group from carbonyl phosphate, and then go to create citrulline, and etc. etc. So this is now going to translocate back into the mitochondria. And those are our 5 steps. So let's look at it again. So remember we started with bicarb, NH4, and 2 ATP for the first step. 4, remember the co-mnemonic carbamyl phosphate synthetase 1. It's important, okay? Which is obligately activated by an acetylglutamate, right? That's going to give us our carbamyl phosphate. Now, carbamyl phosphate is going to combine with ornithine, which translocates from the cytoplasm into the mitochondria, by ornithine transcarbamylase to give us citrulline. Now, these are the only 2 steps that happen in the mitochondria. After that, citrulline is going to go into the cytoplasm, is going to combine with aspartate and create argininosuccinate by our argininosuccinate synthetase enzyme. Okay, and now argininosuccinate, we're going to break it down into arginine by argininosuccinase. And it's going to give us fumarate, which is going to be our connection to the TCA cycle. We'll see that in a second. Now, we get arginine, and we're going to just break it down one last time. Okay, breaking down arginine into urea and into ornithine. Okay, and again, ornithine is going to start the whole process back up again, and that's why it's called a cycle. So in general, there's no net gain loss of, or a net gain or net loss of ornithine, citrulline, argininosuccinate, or arginine, because they keep turning into each other. So they keep cycling back. Okay, and so for this whole process, we have 4 high energy bonds that are used to synthesize 1 molecule of urea, right? Because we needed 3 ATP, and then that gave us 2 ADP. You can see that here as well. So if we go back through the steps, so you can see that here we use 2 ATP, right? Not release the phosphate. Here we used 1 ATP, that also used this, and then this was broken down. Okay, so just so it's clear for everyone. So that's where all of these molecules came from. So we got urea, we got fumarate from here, we got 2 ADP from this step. Okay, we got 2, that gave us 2 inorganic phosphates, and here we got AMP and 2 inorganic. Is that clear for everyone? Just, to really ingrain it in your minds, we're going to go through the mnemonics one more time. So how to remember the urea cycle in the intermediates. We said ordinary careless croppers are also furious about public carriage. So ornithine for ordinary, careless for carbamyl phosphate, citrulline for croppers for citrulline, R for aspartate, also for argininosuccinate, furious for fumarate, about for arginine, and urination for urea. Okay, I hope that cleared everything up. And then again, how to remember the enzymes. We have our cola mnemonic, and just keep this in the back of your head, this delicious looking cola bottle. Carbamyl phosphate synthetase for C. O for ornithine transcarbamylase. Again, I'm going to fix this name, so don't worry about that. It was just a typo on my end. A for argininosuccinate synthetase. And then other A for argininosuccinase, and last for arginine. Okay, so that is how you guys remember the urea cycle. Now, is that clear for everyone? And can we proceed to the clinically relevant stuff? Clear? I'm counting on you guys. Inshallah, everything's clear. So remember what I said about the TCA cycle, that fumarate is what connects the urea cycle, which is here, to the TCA cycle, or citric acid cycle, right? So what happens is this fumarate, and you guys should have learned about the TCA cycle in another lecture. I'm not going to go about it in detail, but fumarate turns into malate, turns into oxaloacetate, turns into aspartate, and now this aspartate can again turn into argininosuccinate. So you can see how these 2 cycles are connected and how they're fueling each other forward. Okay, and again, subhanAllah, the body is interconnected in ways beyond what we can learn, in a lifetime, subhanAllah. So seeing that is very interesting. So again, our argininosuccinate, right, by our arginine or succinase enzyme is going to be broken down into arginine and fumarate. And so this fumarate connects us to the citric acid cycle. Okay, so that can give us malate, oxaloacetate, and then convert to aspartate, which can then again give us arginine or succinate. So you can see the big connection here and the big connection here. So they're fueling each other in order to get rid of this urea or get rid of nitrogenous waste products as urea, which can then be excreted in the urine. Okay. So now I might have to change these colors. I hope it's clear. So now what I want you guys to remember is that the overall control of the urea cycle is by enzyme levels. Okay. So let's say you have someone who has a very, very high protein diet. So the body is subhanAllah going to adapt to that and create more of these proteins that are going to break them down. So we're going to have more enzyme levels, okay, that are related to the urea cycle because you're consuming high protein. So think of like, let's say, bodybuilders, for example. And then second to that is the flow of compounds through the urea cycle also depends on the concentration of the cycle intermediates. So what does that mean? If you have depletion of one of the intermediates for one reason or another, let's say you have a deficiency in one of the enzymes, let's say you didn't eat protein that day, so the cycle is not going to move as fast, okay, or it's not going to move at the same rate because the intermediates in those cycles are not going to be needed. Or let's say your body might be using them for another process. So again, the speed at which this cycle is going going to depend on the concentration of the intermediates, okay, or the molecules that are being made. And again, I cannot emphasize this enough, and this can easily come as an MCQ for you guys, so keep it in mind. The regulation of carbonyl phosphate synthetase 1, I might remember I should have put 1, is via an glutamate. Okay, and now we're going to go through the deficiencies that you can see with the urea cycle, but I just want you to remember that ornithine transcarbamylase is the most common disorder of urea. Okay, so that's the most common problem that we can have in the urea cycle. And remember, ornithine transcarbamylase is going to combine our carbonyl phosphate and ornithine to give us citrulline. Okay, clear so far? This part kind of looks scary, but we're gonna explain it inshallah and make it as easy as possible. So we said we're making urea, right? Why are we making urea? To get rid of ammonium in the blood. Why? Because ammonium is toxic. Okay, so if you have a defect in the urea cycle, what's gonna happen? You can't create urea. So when you can't make urea, what's gonna happen is you're gonna have high levels of ammonium in the blood, right? And that's also and decreased blood urea nitrogen. Why? Because you don't have urea. This is the way the kidneys excrete urea. I think you guys learned that in, because you're teaching renal now. So the defect in the urea cycle would result in hyperammonemia. Okay, so high levels of ammonium in the blood, elevated glutamine, we'll see why in a moment, okay, and decreased blood urea nitrogen. And so these deficiencies are not something that present later in life. Remember, they're genetic defects. If any of you guys attended my previous small pals in pre-med, you remember I told you, when you have, not all the time, but most of the time, when you do have a genetic issue, that's going to present in childhood, like specific deficiencies and defects. Some things can present later, but you guys are going to learn about those. So in this case, during the 24 to 72 hour postnatal period, so after a baby is born, they can have symptoms of lethargy, so very, tired, vomiting, hyperventilation, because they're trying to get rid of all the toxic waste products in the blood. And if it's untreated or undetected, it can progress to coma and death. And so let's just take a look at this model here. So this is the whole urea cycle, right? So let's say we have two deficiencies that we're going to talk about. Well, actually more, but the two main ones, we said OTC or ornithine transcarbamylase deficiency is the most common. So let's start with this. If you can't make ornithine transcarbamylase, if you can't create citrulline from ornithine transcarbamylase combining carbonyl phosphate and ornithine, if that doesn't happen, then every single step here in the urea cycle is not going to happen, right? Because this is the main pathway taking it out from the mitochondria into the cytoplasm and continuing this cycle. So it's not going to happen whatsoever. So you'd expect all of the levels of citrulline, ornithine, arginine, urea to all be decreased. Okay? But the key thing here is that carbonyl phosphate, if it's not used in the urea cycle, it's connected to another cycle that forms something called orotic acid. Okay? So if you have a deficiency here, you're going to build up a lot of carbonyl phosphate. And what's that carbonyl phosphate going to do? It's going to go down this pathway and form our orotic acid. Okay, so what you'd have is high levels of orotic acid and uracil. Okay, you're going to have hyperammonemia, high levels of glutamine, and decreased blood urea nitrogen. Okay, so that's what you'd see in OTC deficiency. However, let's take it a step earlier. Let's say we have our carbonyl phosphate synthetase 1 enzyme, right? The first step that we talked about. Let's say this is deficient. If this is deficient, then everything else is also going to be decreased. So, but the difference is you don't have carbonyl phosphate to create orotic acid. So that's a very key differentiating factor between carbonyl phosphate synthetase 1 deficiency and OTC deficiency, because here you're going to have high orotic acid, right? Because this builds up behind. And if you have deficiency here, you're going to have very low orotic acid because you can't get to that pathway in the first place. Okay? And so when we look at these patients, what can we do to help them, or what can we do to treat them? So what we can do is we can prescribe them a low protein diet. And so they have specific, since they're babies, you know, they can't have, say, breast milk, so they have very specific formula. You know, when you guys mix like the powder and the water, and that's what they use as formula. And so when you have a low protein diet, remember what we said earlier in these principles? Right here. That, you know, the flow of compounds through the urea cycle depends on the concentration of intermediates. So if you're consuming a very low protein diet, you won't have as many intermediates. That way, these patients will not have as severe of a problem as they would if they were just eating very high amounts of protein and whatnot. Okay, so again, what do we treat them with? Low protein diet. Is that clear? And again, just to re-emphasize, and this is from your lecture as well, the difference between carbonyl phosphate synthetase and deficiency and ornithine transcarbamylase deficiency. So both of them are going to have high ammonia levels. You're going to have decreased blood urea nitrogen. You're going to have high blood glutamine levels. But the symptoms are going to be the same. But again, the main difference is that carbonyl phosphate synthetase is not going to have orotic acid urea. You're not going to have high levels of orotic acid, but in ornithine transcarbamylase deficiency, you will. And another difference is that ornithine transcarbamylase is excellent recessive, so it would be more commonly seen in males. And carbonyl phosphate synthetase is autosomal recessive, so you can see that in males and females. Okay, so I hope that makes sense to everyone. But that's again very important, so I want you guys to keep that in mind. Is that clear? Again, so ornithine transcarbamylase, orotic acid. Just remember ornithine for orotic acid. Okay, carbonyl phosphate synthetase, no orotic acid. Okay, so now we're going to move on to talking a bit more about why you have ammonia toxicity. And we keep saying there are high glutamine levels there. What does that mean? So you have two types of treatments that can minimize. So there are two types of treatments that can minimize the toxicity of excess glutamine, which can have effects on the body. Okay, so you have something called phenacetate. Okay, so phenacetate, when you give it to these patients, what's going to happen is it's going to be converted into phenacetyl-CoA. And the important thing about phenacetyl-CoA is that it can take these glutamine molecules, okay, and give you something called phenacetylglutamine. Now, phenacetylglutamine can be excreted by the body safely and more safely than glutamine can. Okay, and why do we care about high glutamine levels? Because they can predispose to brain damage, and let's say these proteins, these molecules leak into the brain, and where they go, water will follow. So that can cause swelling of the brain, which can cause a lot of problems and mental defects and difficulty breathing, and all of these things that, of course, you don't want in these patients. And so, again, we can give them phenylacetate, which turns into phenylacetate-CoA, and that combines with glutamine to give us phenylacetylglutamine, which is excreted by the body. Okay. Similarly, as well, we can get rid of the glycine in the body. Okay. And again, glutamate and glutamine are via this pathway, and glycine is also an important cofactor in this whole process. So when we supply benzoate, it's a very similar process or similar principle. So benzoate becomes benzoyl-CoA that combines with our glycine and gives us hippurate. Okay, it's just another name for the molecule. And hippurate can be safely excreted by the kidneys. Okay, so again, 2 treatments for ammonium toxicity. We can give phenylacetate to remove glutamine as phenylacetylglutamine. And we can also give benzoate to remove glycine as hippurate. So just remember phenylacetate for glutamine and benzoate for glycine. Okay, I hope that. Because this could come as an MCQ for you guys, so that's why I'm just going to keep it in mind. So don't mix up the two. And now our last deficiency for today is going to be our argininosuccinase deficiency. So argininosuccinase, remember, is going to be the third step, right? So we have, so remember, first we have this carbonyl phosphate from CPS1 enzyme combines with ornithine to give us citrulline, and then citrulline and aspartate via argininosuccinase. Synthase is going to give us argininosuccinate, and then you have argininosuccinase to arginine, etc. I'm just repeating the whole cycle again. So here we're looking at this step, breaking down argininosuccinate into arginine and furanate. So the argininosuccinase deficiency. Okay, so here what we have to do is we have to treat with a low protein diet that's high in arginine. Okay, why would we have a diet high in arginine? Why would that make sense? So arginine is going to be important for us to convert this into urea, right? To convert the protein into urea and keep the cycle going. So the arginine is going to stimulate the formation of N-acetylglutamate, which remember we said is very important. And that's going to activate the urea cycle to get rid of excess ammonia and excess glutamine. Okay, so here just remember, for argininosuccinase deficiency, we need to treat with a low protein diet that is high in arginine. Why? What happens is the argininosuccinate is going to be excreted in place of urea in the body, right? Because you basically can't get to this step with urea. So you want a diet that's going to be high in arginine, okay? And low in protein, so that this can be excreted. Why is it high in arginine? Again, because that's going to stimulate the formation of an acetylglutamate, which is the key for this step here. You can see it. Okay. And that's going to get rid of excess ammonia in the blood. And now this is just another figure to make sure it's clear for you guys. So we have, again, the mnemonic ordinary careless crappers are also curious about public urination. And for the enzymes, it's going to be cola. Okay, so we're going to get, I'll repeat your point once again. So we're going to start with our carbonyl phosphate synthetase. So combining all of the predisposing molecules by CPS1 activated by N-acetyl. N-acetylglutamate. Okay, and that's going to give us a carbonyl phosphate. That's going to combine with ornithine to give us ornithine, by ornithine transcarbamylase, to give us citrulline. Citrulline goes out, is going to give us argininosuccinate by argininosuccinate synthase, and it's going to combine with aspartate to do so. And then we're going to break argininosuccinate down to arginine and fumarate. Arginine is going to be broken down into ornithine and giving us urea, which is excreted safely in the. I'll go back to your point. Okay, so when we say that's the result, now we have maybe, let's go, maybe this picture would be better. Okay. So arginine is very important for N-acetylglutamate. So the higher arginine levels you have, the more this is going to become active, right? And we know this is the key step in feeding the urea cycle forward. So when you have arginine succinase deficiency, what ends up happening is you can only make arginine succinate and you're going to stop here. Okay, so the process can't continue to make urea. However, arginine succinate can still be excreted in the body. So we want a low protein diet to minimize the amount of urea we have to make, but we want it to be high in arginine in order to feed the cycle forward. So you're basically just pushing it out this way. And that's why that's the treatment for arginine succinase. Did that answer your question? I'll show you again here. So again, we have arginine, okay? Usually it's going to give you urea, and that's going to excrete it outwards. So I'll explain it another way, but I just figure it's much easier to explain them. So if you can't make arginine succinate, or you can make it, but you can't break it down. So what you're kind of doing instead is you're supplementing it here, because this enzyme is working, this enzyme is working, this enzyme is working, this enzyme is working. So you want high levels of arginine to kind of stimulate this to move forward, right? And to push it out this way, because this enzyme is working. So if you have high levels of arginine, then you can still break it down, right? You can still give urea and ornithine, and then the cycle is going to continue out. Does that make sense? Hope that answered your question. Okay, so now, do you guys have any other questions? I hope the urea cycle was relatively straightforward. It's one of, I'd say, the easier lectures. It's just you have to make sure the names are distinct, and I hope the mnemonics helped with that. Now I have a bunch of questions. You guys don't seem very active for this lecture, but it's fine. So I'm gonna, break them down and go through them, because these are, when I did MOL and I do this PAL in first year, the questions I put were the exact same questions that came in our exam. And a lot of them were from Lippincott, a lot of them were from USMLE Step 1, because this specific lecture was taught by Dr. Ahmed Al-Jata. So right now, so let's read this case. So a newborn becomes lethargic and drowsy 24 hours after birth. Blood analysis shows hyperammonemia coupled with erotic aciduria. This individual has an enzyme deficiency that leads to an inability to directly produce which of the following? Okay, so first let's take it step by step. We saw that this is a lethargic baby that's drowsy 24 hours after birth. So we're thinking of a genetic defect, okay? Okay, good morning. So the important thing is, or the key thing in this question is they told you, sorry, there's erotic aciduria. And remember, erotic for ornithine transcarbamylase. So this is an ornithine transcarbamylase deficiency. This question is indirectly asking you, what is the product of the reaction that OTC catalyzes? And you guys are correct. It is formation of citrulline. Excellent. And so again, the same vignette. A potential treatment for this patient is supplementing which of the following? This one might be a bit tough because this was a repeated question, but what I want you guys to choose from, ignore the first two. I want you to choose between C, D, and E. Okay, I know it was a confusing question, so I'll walk you through. Now, if we're looking at the urea cycle again, arginine is one of the intermediates there. Okay, so arginine, you guys are right. So if you supplement with arginine, you might be able to help the urea cycle move forward. Remember, it's the same principle that I explained when I'm asked this question. So benzoate, remember, what does that do? Benzoate is going to combine with glycine, and it's going to be excreted as hippurate, which can help lower the levels and improve the symptoms in this patient. Lysine has no specific role. Glutamine also has no specific role. Phenylbutyrate, glutamine could actually worsen the patient's condition. Phenylbutyrate can be given. Remember, phenylbutyrate combines with glutamine, and that's going to give us phenyl. The thing about MOL is that the names are not here. Oops. Sorry, that was the trick question. It wasn't phenylbutyrate, it was phenylacetate. So don't make that same mistake. But yes, the correct answer was C, benzoate. So remember, you can give benzoate that's going to be excreted as hippurate, or phenylacetate that's going to be excreted as phenylacetate glutamate. Okay. But you guys were right in both of them. All right, there's a lot of questions, so I'm just gonna, go through them. If you guys want to answer, feel free to do so in the chat. So a female neonate did well until approximately 24 hours when she became lethargic. A sepsis workup was negative, so there was no bacteria in her blood. At 56 hours, she started showing focal seizure activity, so that tells you there's problems with the brain. Plasma ammonia level was found to be 887 micromoles per liter, which is very elevated, so very high ammonia levels. And quantitative plasma amino acid levels revealed a marked elevation of citrulline, but not argininosuccinate. Okay, so what do you guys think the deficiency is in? Okay, yes. I don't know why all of these are C for some reason, but if you're stuck, I guess choose C. But yes, so again, they're basically telling you high levels of citrulline, but low levels of argininosuccinate. So why? We have argininosuccinate synthase deficiency. Okay. Which of the following would be elevated in the blood of this patient? Same patient, same case we just talked about. Yes, you guys are right. So B will be elevated. Remember, glutamine is one of the things that causes problems, causes the CNS symptoms that we can see, and can contribute to cerebral edema or swelling of the brain. And so remember, that's why what binds to glutamine is the phenylacetate, that's converted as phenylacetate. The one that I went back to the side. The names are annoying, I know, but yeah, so this is glutamine. And a trick in this question would be urea. Urea is not elevated. Remember, you cannot make blood urea nitrogen, okay, in these specific scenarios, which is why you supplement with later steps like arginine. Okay, so again, just to answer this question, this is to re-emphasize the point that Omar asked about. So why might supplementation with arginine be of benefit to this patient? So remember, if you don't have arginine and succinate synthase, the step that is going to give you urea is arginine, right? Broken down into urea, so arginines. Okay, so what happens, or the key part here, is you're basically bypassing this part and you're supplementing arginine to push the cycle forward. Yeah. So that's the explanation here. Last couple questions. I don't know how many we have left, but I'm just going to keep going until they're done. Infant born to a 22-year-old female is diagnosed with inherited condition that results in impaired transport of ornithine from the cytosol of the mitochondria. Okay, so impaired transport of ornithine. Restriction of which of the following substance in the diet can improve this patient's condition? This is basically going through the concept of the whole thing. So if you cannot transport ornithine from the cytosol into the mitochondria, that means you cannot do the first two steps of the urea cycle, right? And so because of that, you can't make your citrulline and you can't feed forward the urea cycle. So it's kind of just asking you, how do you treat in general urea cycle defects? Or what can you restrict in the diet? Proteins. Oh, perfect. You guys are great. And I remember this question very well. This came in our TBL after I gave this power. Allah, I was so happy when that happened. Anyway, oops. I revealed the answer. Pretend you guys didn't see that video. Oops. Exposing myself. Anyway, infant born to a 32-year-old female demonstrates lethargy, vomiting, and seizures after the first initial feeding. Initial laboratory evaluation demonstrates a markedly increased blood ammonium level. Remember, blood ammonium, not urea. Okay. Liver biopsy suggests impaired formation of N-acetylglutamate. And N-acetylglutamate is important for what? Can you guys tell me what the enzyme is? Not OTC, it's CPS1. Just keep that in mind. OTC, the only like activator that you have is for the first step, which is CPS1. Okay. Ahmad, you're talking about this question. Let me go back. Okay, so this is basically telling you that there's a defect where ornithine cannot be transported into the mitochondria. And if you remember, the first two steps of the urea cycle happen in the mitochondria. So if you can't do the first two steps in the urea cycle, that's going to decrease the amount of urea your body is able to make. And so remember, we said the general treatment principles for urea cycle defects is to restrict protein. Because remember, the more protein you eat, the more the cycle is going to go forward. So if the cycle is not able to work properly, what happens is you're eating a lot of protein, but you can't excrete it safely in the body, which creates a lot of nitrogenous waste product. So that's why for these patients, we usually restrict protein and we can supplement RP. That's okay. Unless you guys got right. Okay. Next, a 2-year-old male demonstrates periodic lethargy, vomiting, and confusion. Laboratory testing reveals increased blood ammonium levels during these 3 episodes, as well as marked urine orotic acid excretion. Okay, which of the following enzymes is most likely to be deficient? Remember, orotic acid is for ornithine transcarbamylase. Perfect. Alhamdulillah. Okay, amazing. And remember, if you have carbonyl phosphate synthase 1 deficiency, then it is low in orotic acid. You don't have orotic aciduria. You are just going to have a buildup of the other products, like glutamine and whatnot. Okay, we're done with the first lecture. Do you guys want like a 10-minute break? If you guys want to go eat. Asr is still in a bit. Let me check what time is Asr exactly. Okay, Asr should be now. So if you guys want to come back in like 15 minutes, whoever wants to pray, whoever wants to grab a snack, and then inshallah we'll finish the last lecture. Does that sound good for everyone? Oh, that's perfect. We'll be back at 3:13. That's perfect. Okay, so since it's going to be a separate recording, again, Assalamu alaikum everyone. My name is Maryam Altayeb, and today we're going to be learning about amino acid degradation and synthesis. Again, if you guys have any questions, please stop me at any time. I'm always here and ready to answer. I would just say, I apologize because this is the first lecture I've given where I have not made the PowerPoint. I instead took Dr. Rajat's PowerPoint since it was already long, and I just will be explaining on it. So at least that'll be easier for you guys because you have the same slides to reference. And I tried to add some pictures here and there, some mnemonics that I found helpful. And inshallah you find them helpful as well. But please, I apologize for the design. And the graphic designer inside of me is currently dying. This Spanish. You gotta do what you gotta do, I guess. Okay. And start. Oops. Can you guys see my screen? Or which screen can you see? Can you guys see the PowerPoint without the notes part, right? On this one. Okay, that's great. All right. Just so you guys can see the full screen. So we're going to be talking about amino acid degradation and synthesis. Now, I just want to preface this by saying this is lecture 15 and 16 in your syllabus, but I rearranged the slides. I want to talk about synthesis first and then degradation because that only makes sense in my head, you know, for you to understand how amino acids are made and then how you break them down, you know. So let's give that a shot. Okay. So now we're going to talk about the synthesis of amino acids. So there's 3 main processes by which we synthesize amino acids. There's something called transamination. So amination, when we talk about an amine, that's any nitrogen group, right? So transamination is transferring an amine group from one amino acid to another in order to convert them. So for example, you can make alanine from pyruvate by a transamination reaction, okay, of a keto acid. Amidation is similar, and we're going to explain like the specific differences between them, and then you can just synthesize one amino acid from the other. For example, tyrosine can be made from phenylalanine. Okay. Wait, give me 1 second. I want to make sure there are no. I apologize. Sorry. I just want to make sure that I hate having to press the narration animation. Sorry. Let's start again. My bad. So now that's how you synthesize amino acids. But in order to know which amino acids we can and cannot synthesize, we have to know that there's different types of amino acids. There's essential and non-essential. Essential means you cannot obtain them from the diet. You have to get them from essential means you have to obtain them from the diet, okay? You cannot create them in the body, or you cannot synthesize them de novo, okay? And there are all 20 types of amino acids that I'm not going to be going through today, okay? And so, like I mentioned, essential amino acids cannot be synthesized in humans and must be provided from the diet, okay? Sister is not feeling well, I think. Okay. The only exception is that arginine can be required during specific periods of growth or positive nitrogen balance, such as pregnancy. And just remember from the other lecture, arginine can be supplemented as well in some urea cycle defects. Okay. And so we know there are 20 types of amino acids in total. Of those 20 types, we have essential and non-essential amino acids. So our essential amino acids we're going to talk about, and I have a mnemonic for you guys to remember them. And then you have non-essential ones as well. So non-essential means you can make them in the diet. Essential means you have to obtain them from the diet. Okay. So non-essential, you can make them in the body. Essential, you need them from the diet. And so this is a mnemonic. This is the one that we've been learning since first year for our time. And there's another one I'm going to add as well in case you guys don't like this one. So the essential amino acids are private Tim Hall. So PVT, Tim Hall. So P for phenylalanine, V for valine, T for threonine, okay? Other T for tryptophan, I for isoleucine, M for methionine, and then Hall. So H for histidine, A for arginine, L for leucine, and other L for lysine, okay? So remember, isoleucine, leucine, lysine, they're easier to remember, arginine, histidine. However, again, for arginine, it's semi-essential, like we mentioned in the last slide, meaning that you need it in stages or periods of growth more than you do in general. So you need it more specifically in childhood, pregnancy, and positive nitrogen balance states. And again, this is the same mnemonic. And we're going to talk about glucogenic, ketogenic, and ketogenic in a bit. Okay, so, but just keep in mind that you have essential and non-essential amino acids, and from those you can subclassify them. Okay. So now we're going to talk about the synthesis of specific types of amino acids. And when we talk about synthesis of amino acids, that means in the body. So these are non-essential. You don't need to obtain them from the diet, like the ones in the mnemonic private Tim Hall. And so, for example, non-essential amino acids are synthesized from intermediates of metabolism, like we just said. And so, for example, tyrosine is made from phenylalanine. This is an important example for you guys to remember because it can come up in some questions. Cysteine is made from methionine, and this we're going to talk about in a bit in specific detail. Okay, but just know that we're going to be talking about how you create specific types, or synthesize specific amino acids. Okay. And there's different ways in which you can synthesize them. There's transamination, reductive amination, amidation, conversion, synthesis, and we're going to talk about all of these now. Okay. So transamination, like I told you guys, that's when you're transferring an amino group. Okay. So for example, you transfer an amino group to form an alpha keto acid, and your end product. So pyruvate can be converted into alanine, okay, by an aminotransferase enzyme. Oxaloacetate can be converted into an aspartate, and so these require an alpha ketoacid, okay? And then you can have alpha ketoglutarate converted into glutamate. And do you guys remember where you can see this? Where this reaction is from? No, maybe. It's fine. But I'll wait for you guys to answer that later. Okay, so these are transamination reactions. These are transamination reactions. Okay, so you're just converting an amino group from one to the other. Okay, then you have something called reductive amination. Okay, so reductive means you're removing something, you're reducing it. So, and the exception to this, or the specific important amino acid for this, is glutamate. Okay, and it's written glutamate is unusual and also being synthesized by the reverse of oxidative deamination. Okay, catalyzed by glutamate dehydrogenase when ammonia levels are high. Okay, so what does that mean? You have something called, so you guys remember in high school you learned about redox reactions, okay? And so specifically what happens with glutamate, you can do both. You can make it by transamination and you can also make it by reductive amination. And what's the difference between one and the other? Reductive amination is the reverse of oxidative deamination, okay? And so what happens is, so this is transamination, right? So you're gonna switch the NH groups, okay, by aminotransferase enzymes. Oxidative deamination, okay, is going to lead to disposal of your NH3, okay? That's in disposal. And synthesis, which is what we're talking about now, is reductive amination. So you're taking it and you're putting the amine group here. Okay, that's going to convert alpha ketoglutarate and by different processes and convert it to glutamate. Okay, so you see how it's reversal. So glutamate can turn into alpha ketoglutarate by transamination, and then alpha ketoglutarate can turn back into glutamate by reductive amination. Okay, by glutamate dehydrogenase. So this is just a specific example. More eliminations. I'll ask you guys later a question of this alpha ketoglutarate to glutamate, but I just answered it. So, okay, so synthesis by amidation. Okay, in amidation, we're adding a secondary amide group. Okay, so why is that different? So you have glutamate and glutamine, and they're not the exact same amino acid, they're different. So glutamate is here, it just has one NH group, and then you can convert it into glutamine, and it has two. So this is amidation, okay, and this is going to be amination. So you can see how this is a charged molecule, and this is incorporated into the molecule as well, okay? So that would be the difference between, let's say, glutamate and glutamine. Aspartate and aspargine, aspartate and aspargine, okay? So this is by amidation reaction. So why are we calling them amides, not amines? Because they're carboxylic acid derivatives where you don't have an OH group that is replaced by the NH2, okay? So this OH group is replaced by an NH2. So again, glutamine is produced by amidation of glutamate. Aspargine is produced by amidation of aspartate. Okay, so this is the difference between these two molecules. Okay, now we have glycine and serine. So glycine and serine, if you guys remember, glycine is the only amino acid that has two of the same side chain, right? It has two H's, it's the smallest one. And so you can convert it to serine by something called serine hydroxymethyl So let's just look at the name. So serine hydroxymethyl transferase. So you're transferring a hydroxymethyl group from something in order to turn glycine into serine. Okay, and that's going to require something called, methyl ethylene tetrahydrofolate, which we're going to talk about more a bit later. and that's going to give us tetrahydrofolate. Okay, and why do we need serine? Because serine can be then converted into pyruvate. And we know pyruvate is important for glycolysis and all other metabolic processes in the body. So again, we can make serine from glycine, and the difference between both is a methyl and a hydroxyl group, hydroxymethyl. That's why it's, the names in biochemistry, as long as they do make sense most of the time. And so it's going to add the hydroxyl group to glycine to turn it into serine, okay? And the donor of the methyl group is what? Is going to be methylene tetrahydrofolate. You're going to remove the methyl and it's going to give you tetrahydrofolate as a reaction, okay? So you need this in order for this reaction to occur properly. And these are all derivatives of folic acid or vitamin B9. And again, we're going to talk about that later. But just so you guys can see, serine is going to combine with tetrahydrofolate to give glycine and, I'm just going to say MTHF, and this reaction can go the other way, right? So glycine and MTHF can convert into serine and tetrahydrofolate, okay? Now we're going to talk about, again, the interconversion of serine and glycine. So we said serine is formed from 3-phosphoglycerate as an intermediate in glycolysis, okay? And like we mentioned in this previous slide, so serine can be formed via glycolysis, okay, as an intermediate, and serine can also be formed by glycine, okay? And that's the reaction we just exactly said. You don't need to know this reaction. I'm just mentioning it as an example so you guys know that, what are the different ways that you can create amino acids. So there can be interconversion of serine and glycine, like we mentioned here, and serine can also be converted from glycolysis. This is, again, the reaction that I just explained in the picture. Okay. Now this, I want you guys to pay attention to because it is actually important for you guys to know because it's going to have some clinical correlation that we'll talk about a bit later towards the end of the lecture. So I want you guys to just focus with me for this point and keep it in the back of your mind, and we'll come back to it later and I'll re-explain it. Okay. So remember we said the mnemonic for our essential amino acids, private Tim Hall, and the M in Tim is methionine. So methionine is one of our essential amino acids, okay? And so why do we need methionine and why is it important? Okay, well, it can form cysteine, okay? And cysteine can then be used to form homocysteine and other molecules, and those are going to require specific vitamins. And again, I'll explain this in detail later on, okay? But you can only create cysteine if you're taking enough methionine in your diet. If you're not adequately taking in your amino acids, then this process cannot happen, okay? And what happens is you have homocysteine is going to combine with serine and form cystathionine. And cystathionine is going to be converted or hydrolyzed to alpha ketobutyrate and cysteine. So this is what we're looking for, okay, our cysteine molecule. And alpha ketobutyrate can be used in other intermediate processes in the body, okay? And again, this whole process is going to require your different vitamins. It's going to require vitamin B6 here and here, and here it's going to require vitamin B9. But again, we're going to talk about that a bit later. Okay, for now, what I want you guys to know is that it's more easily shown here. You need methionine. Okay, I'll talk about this process later, so don't worry. Methionine in order to make homocysteine, and homocysteine makes sense that that's going to make cysteine. Okay, and you need cysteine for different metabolic processes in the body. Okay, and so why are we talking about homocysteine, cysteine? What do all these mean? Well, we said methionine can be a source of homocysteine. And the problem with homocysteine is that it's been linked to specific vascular diseases. So if you guys know what atherosclerosis is, we're having fatty plaques in the arteries. So that's going to block blood flow. And this is a very important pathological process of things like heart attacks or myocardial infarction and different things like that. And so plasma homocysteine levels are inversely related with folic acid, B12, and B6. So remember, I said we need folic acid, vitamin B12, and vitamin B6. So if you don't have these, okay, then you're not going to be able to properly convert homocysteine into cysteine. and having excess levels of homocysteine in the blood is linked to specific cardiovascular diseases and vascular diseases in general. Okay, again, I'm going to explain this later on in the lecture. I just want you guys to keep it in the back of your mind. Okay, so incorporation of homocysteine, which we were just talking about, into a protein can be via those disulfide bonds. Okay, remember with sulfur binding to another sulfur. Okay, and this disulfide bond is going to cause like a fold within the protein, and that is going to cause alterations in structure and function. And so another link of homocysteine or homocysteinylation is that it's linked to toxicity, autoimmune responses, which might be the reason it contributes to atherogenesis. So formation of this atherosclerotic plaque, which is bad for the body, can cause heart attacks, vascular disease, etc., etc. Things you're going to learn about, inshallah, later in that school. Okay, but I just want you guys to understand right now that we need methionine in the diet. Okay, and this process can only go if you have methionine and you have the adequate vitamins. And methionine can go through this whole process to form homocysteine and then cysteine. But homocysteine can be bad. It can cause formation of vascular disease, atherosclerotic plaques. So how do we lower homocysteine levels? It's shown that higher levels of vitamins, so folic acid, which is vitamin B9, vitamin B12, and vitamin B6, are inversely linked to homocysteine levels. Okay. Now, again, related to homocysteine is something called folic acid deficiency and vitamin B12 deficiency, but specifically folic acid is linked to something called spina bifida, if you guys learned about it. So spina bifida is basically when you have an open defect here in the spinal cord. So you guys remember the different layers of mesoderm, endoderm, ectoderm, and so the process of disclosure is not complete. And what happens is the spinal cord of the baby can look outwards, and this is what we call a myelomeningocele, in medical term. And so the disease is called spina bifida. And so what happens is mothers that are not supplementing with these vitamins in pregnancy can lead to brain problems in the baby, can lead to neural tube defects such as spina bifida. And so that's why mothers need to supplement with folic acid even before pregnancy in order to reduce the risk of such defects. So this is, if any of your moms have taken pregnant care, this is a very popular one. And again, it's marketed as something that has folic acid. So it's very important for pregnancy and for preconception as well. I just added this picture so you guys can have that mental association. Okay, now before we go on, you guys have any questions? Again, knowing that I will repeat the whole homocysteine cysteine thing later on. All good? Okay. Okay. So moving on from spina bifida, we can synthesize tyrosine from phenylalanine. Remember, this is one of the things I told you guys was important. And again, mnemonic private Tim Hall, first P is for phenylalanine. Okay, and so phenylalanine is an essential amino acid. You cannot obtain it from, you cannot create it in the body. You must obtain it from the diet. And the reaction of converting phenylalanine to tyrosine requires something called THB or tetrahydrobiopterin. Okay, and so tetrahydrobiopterin is one important element, and the enzyme is called phenylalanine hydroxylase. And the key clinical correlation here is something called phenylketonuria. So when there's a deficiency in this enzyme, you cannot convert phenylalanine to tyrosine. So levels of phenylalanine are going to build up, and that can be, again, can have its toxic effects in the body. And the key thing here is that these patients cannot have aspartame. And aspartame is found in specifically sodas. They use it in a lot of artificial sweeteners. So these patients really need to stay away from that because that can, increase your levels of phenylalanine because aspartame is made up of phenylalanine. Okay, so that's why I added this aspartame free. Okay, because these patients that have a deficiency, PKU, phenylketonuria, cannot consume aspartame because phenylalanine levels will get too high and it's going to be toxic in the body. Now, let's review this part. These are again the same questions that Dr. Raja answered. So we just talked about Okay, so match the amino acid to the intermediate needed for its synthesis. Oxaloacetate, let's start with B. Oxaloacetate can be converted into what? Propylene. Okay. So alanine is made up of, like you guys said, pyruvate. Okay. Oxaloacetate can be used to make aspartate. Okay, or aspartate is made of oxaloacetate. And then we have glutamate is made up of alpha ketoglutarate. I always remember this because the names sound similar. Alpha ketoglutarate, glutamate, it makes sense that they're linked. Okay. But again, you guys need to know all of the intermediates and what they can be converted. Okay, so now that was the first part of the lecture. How do you synthesize amino acids? Now we're going to look more towards, okay, now you have these amino acids. What's going to happen to them? What can you do with them? So we briefly mentioned it, but now remember how we said breaking down proteins, breaking down amino acids is going to lead to, that's what fuels the urea cycle forward. That's why you can restrict protein in their diet. So after ammonia has been removed from the amino acid, what happens to it? There's this carbon skeleton that remains, and you need to understand what are the different things that can be done with that. Because like we said, amino acids can be interchangeable. One amino acid can be created into another amino acid, and etc., etc. Okay, and these can fuel different metabolic processes in the body, even such as glycolysis and other things. Okay, and used to make other compounds. So we generally group them into two groups. So you have the amino group, which is a nitrogen. The nitrogen can be incorporated into urea. So this is via the urea cycle and then excreted. And then you have your carbon skeleton, which is an alpha keto acid. You can use it to synthesize other compounds, or you can break it down to make energy. And so, again, like we said, removal of the amino group and metabolism of carbon skeletons. Okay? And now, these 7 products, the reason they're important is because we said we can use these amino acids for energy. Now, there's different forms of energy. You can break down energy in terms of fats, and you can also break them down in terms of sugars, like glucose. And so, you have specific things like acetyl-CoA and acetoacetyl-CoA and their derivatives are classified as ketogenic because they can be used in the process of ketogenesis. Okay, and those provide energy from lipids usually as a source. And then you have other products such as, which are the ones we commonly heard about, pyruvate, oxaloacetate, alpha ketoglutarate, succinyl-CoA, fumarate. Remember, fumarate was the link between urea cycle and TCA. Okay, and so these are glucogenic. Okay, and so, for example, pyruvate can give glucose by gluconeogenesis and by the other intermediates in the Krebs cycle. Okay, so now that we know that there are ketogenic and glucogenic amino acids, some of the amino acids can be both ketogenic and glucogenic. Okay, and so when we say they're glucogenic amino acids, that means breaking them down, their catabolism produces pyruvate. Or if not pyruvate, any of the intermediates of the citric acid cycle that we talked about, right? Alpha ketoglutarate, succinyl-CoA, fumarate, etc. And the key thing here is that all of these can be converted into glucose. That's why it's called glucogenic, meaning it can make glucose. Then we have ketogenic amino acids, okay? Can be used to make ketones, remember ketogenesis, okay? And so ketogenic amino acids are when their breakdown produces acetoacetate, or one of its precursors, which are the ones we'll normally hear about, acetyl-CoA or acetoacetyl-CoA. And why are these important in ketogenesis? Because they can be converted into fatty acids and ketone bodies. Okay, and there's this mnemonic here. Okay, these are looking at the 9 essential amino acids. Okay, and so this is another mnemonic if you guys like it. My tall, handsome vegan friend is watering kale leaves, but I'm very used to private Tim Hall, so I'm just going to keep using that one. But the key thing here is that this is showing you which amino acids are ketogenic, which are glucogenic, and which are both. So the only exclusively ketogenic, meaning they're only there for ketogenesis, are lysine, leucine, the 2 L's, easy to remember. Lysine, leucine, always together, and they're essential. Okay, the exclusively glucogenic ones are methionine, histidine, and valine. Okay, methionine, histidine, and valine. And ketogenic and glucogenic both are, the mnemonic is don't fit. So the F is really pH, so phenylalanine, isoleucine, tryptophan, threonine, and tyrosine. That's the extra T. Okay. Does that make sense for everyone? Again, this is just an easier way for you guys to remember it. The way I usually memorized it was, I'll show you guys this table. Just remember, exclusively ketogenic is lysine, leucine. Okay? Glucogenic are most of them. So what I used to do is I used to memorize ketogenic, and the ones that are both glucogenic and ketogenic, and the ones that are only glucogenic is everything else. It's much easier to exclude that way. So ketogenic is leucine, lysine, okay? Both glucogenic and ketogenic is, they don't fit. So the F is for pH, so phenylalanine, isoleucine, tryptophan, tyrosine, okay? Which is this number. All right. So now when we say an amino acid is both ketogenic and glucogenic, what does that mean? So for example, we said fit, so the P is for phenylalanine, for example. We said phenylalanine can be converted to tyrosine, you need THB, right? The effect on this enzyme gives us phenylketonuria. So you don't have to know this whole thing, but just know that throughout this whole process, eventually you can get fumarate and you can get acetoacetate. Okay, we said fumarate is one of the pyruvate derivatives, so this is glucogenic. Acetoacetate, right, like acetoacetyl-CoA, acetyl-CoA, this is one of the derivatives that can be used in ketogenesis. So that's why this is both ketogenic and glucogenic. So you can make something with ketones, you can make something within the TCA cycle. Okay, so that's for phenylalanine. Isoleucine, for example, okay, can be converted into acetyl-CoA for fatty acid synthesis, also can be converted into succinyl-CoA for the citric acid cycle. So again, it's both glucogenic and ketogenic. Okay, as well, tryptophan can be converted into pyruvate. Tryptophan can also be converted into acetyl-CoA, and as well as threonine. Okay, so again, this can go into glucogenesis, this can go into ketogenesis. So again, fit, those are the both glucogenic and ketogenic amino acids. Lysine, leucine, exclusively ketogenic, and all of the rest are glucogenic only. Easy enough? Okay. And again, remember, lysine, leucine can be converted into acetyl-CoA, so they're exclusively ketogenic. And this is again the images that Dr. Raja included for you guys. Now, let's practice. We said lysine, leucine are exclusively ketogenic, right? And we said our mnemonic is fit, so phenylalanine, isoleucine, threonine, what else? Tyrosine, okay, anyway, these ones, tyrosine, tryptophan, okay? So now, lysine, we said this is exclusively ketogenic. Phenylalanine is both ketogenic and glucogenic, okay? And the rest of them, I don't see leucine. So without even thinking, so long as you know lysine, this is ketogenic. This is both ketogenic and glucogenic. By default, all the rest are only glucogenic. So you don't really have to memorize this whole table. If you just memorize these, then the rest is easy to exclude. Okay? Easy enough? Okay, so then answer this question, please. Which of the following amino acids is exclusively glucogenic? So again, we can do it by elimination. Yep, exactly, alanine. Remember, phenylalanine is the F, so that is both ketogenic and glucogenic. Lysine is exclusively ketogenic. Okay. And isoleucine is both. Tryptophan is both. So alanine is the only exclusively glucogenic. Okay. Threonine. Okay, so there's different amino acids, some that are ketogenic and some that are glucogenic. Okay, and I don't know why this table doesn't include threonine, although it's, you know, it's both ketogenic and glucogenic. Um, but so all of these amino acids can be converted into acetyl-CoA, meaning they're ketogenic. Okay, and they can also be converted, at least for the fit mnemonic, so phenylalanine, isoleucine, threonine, tyrosine, tryptophan, into pyruvate as well. So that makes them both ketogenic and glucogenic. And again, the only ketogenic, where's my favorite picture yet? The only ketogenic ones are lysine, leucine. So those are easy to remember. Fit is for the both, and um, the rest, all of the rest are just exclusively glucogenic, which are most amino acids. Okay, and again, for the last time, don't forget these ones. Okay, and again, this is another mnemonic if you guys don't like private MOL for the essential amino acids. But just remember, alanine, valine. All the other sources, including this figure, say that threonine is both converted into pyruvate and converted into acetyl-CoA. Tyrosine is the only non-essential amino acid that is both glucogenic and ketogenic. But مثلا here it says threonine, here it's threonine is classified as glucogenic only. So that's why. Yeah, tyrosine is the only non-amino acid that is considered both glucogenic and ketogenic, because it can be converted into both. You can see tyrosine, and it can also be converted into fumarate. So just ignore that point. But for now, what helped me is memorizing this mnemonic, the ones that don't fit. So phenylalanine, isoleucine, tryptophan, threonine, and tyrosine. So those are the essential, and those are how you classify them. Inshallah, easy peasy. So again, which of these is glucogenic and essential? So remember, our glucogenic and essential, our mnemonic is private Tim Hall, so phenylalanine, valine, threonine, that's Tim. The answer is valine. So remember, leucine is both is ketogenic, isoleucine is both, tryptophan is both, serine is exclusively glucogenic, but it is not an essential amino acid. Okay, so you guys are good. Now we're going to talk about different degradation pathways. So this is kind of similar to the different synthesis pathways. You can either remove an amino group, okay? There's specific metabolism of these molecules. So threonine, methionine, and cysteine, which I'm going to talk about again, and the propionyl-CoA pathway. Okay, so we're just going to go through these now. But you guys understood the first part, and you know how to classify. Just again, from the first part, make sure you know which amino acids give you what, and what are the essential amino acids, what's ketogenic, what's glucogenic, because that's, easy points in MCQs, because they're very common. Again, so this is revision. So some amino acids are converted into intermediates of the citric acid cycle. So we said alanine can be converted into pyruvate, aspartate to oxaloacetate, glutamate to alpha-ketoglutarate, asparagine to aspartate, glutamine to glutamine. And so these are by removal of simple amino groups. Okay, remember we said when you do amidation reactions, so when you're adding an amino group, you're converting it the other way. All right, like aspartate to asparagine, glutamate to glutamine, the deamination reaction is how you convert it reversely. Yes, but let me go back to the slide just so I solidify that for you guys. I'm talking about yeah, so remember, amidation is how you convert glutamate to glutamine, aspartate to asparagine. Deamination is going to be the opposite. So you're removing an amine group instead of adding it. Okay? Let's go back to Yes. Okay. So this is deamination reaction, and so on. And you can do this for various amino acids, but just keep these in mind. And remember that the conversion of these, for the most part, is interchangeable. So here's removing an amino acid. If you add an amino acid back, it goes the other way. So for example, alanine is converted into pyruvate by ALT. And ALT is an enzyme that we can find in the liver, right? And so alanine aminotransferase, that's what it's called. And it's one of the important markers of liver function. So it converts alanine into pyruvate and vice versa. And so when it converts alanine into pyruvate, it's going to give us glutamate. Okay. When it goes the other way, it's going to take pyruvate and glutamate, give us alanine and alpha ketoglutarate. Okay. So these are interchangeable. But the important thing to know is that for the ALT enzyme to work, it requires vitamin B6 as a cofactor. So if you have vitamin B6 deficiency, that means it won't work properly, and that can lead to liver problems along with problems in other places in the body. So for now, the two things we talked about with the vitamin B6 that are important are going to be the conversion of homocysteine into cysteine, right? And now for ALT enzyme to work. So just two things you guys need to remember about vitamin B6. And now asparagine and aspartate are related. The same way we have the alanine aminotransferase, you also have the aspartate aminotransferase, which is also found in the liver and other parts of the bodies. So again, the name makes sense. Aminotransferase. So you're transferring an amine group from one molecule to the other. Okay. And so for asparagine to form aspartate, you're going to transfer the amino group. Okay, so you're removing it here. See how there used to be an amino group here and now it's removed. So asparagine, I think of it like asparagine. The extra N means it has an amino group. Aspartate doesn't, so it's removal. Okay, and that's by asparginase. So it's breaking the amino acid. And then you have aspartate can be converted into oxaloacetate by aspartate aminotransferase. Okay, so just keep that in mind because these are all interchangeable. And so asparginase, this enzyme that breaks this amino acid off to give us aspartate, okay, also forms ammonia. Okay, and an interesting clinical correlation is that this enzyme is used to treat leukemia and lymphoma. Why? Because, subhanAllah, cells in leukemia and lymphoma, which is cancers of the blood, cannot synthesize asparagine. Okay, so if you're going to give L-asparginase, what's that going to do? It's going to break down the asparagine, into aspartate. So now you're depleting the levels of asparagine, which is a very important amino acid, for these cells to survive. And so because of that, it's going to help kill the cancer cells. Okay, so L-asparginase deprives leukemic cells or these cancer cells of asparagine, which leads to cell death. So again, just to recap, we have an enzyme called asparginase, which takes the amino group off of asparagine, leaving out ammonia. And in specific cancer cells, they cannot make asparagine on their own. Normal cells can, but these cancer cells of the blood cannot. So a medication that was created is L-asparginase, so you're supplementing this enzyme to break down asparagine more and more and more and more. And because the cells require this to survive, it's going to cause the cancer cells to die without it. Which is, I think, a very interesting clinical correlation. Understood? Good? Easy enough and prompt? Yes. Okay. So now we move on. Again, we already talked about this. We're going to touch upon it one more time. Phenylalanine and tyrosine metabolism. So phenylalanine is converted into tyrosine via the tetrahydrobiopterin. Okay, so that's very important. And like we said, remember, deficiency in this enzyme means they can't take aspartate. Remember, they cannot drink soda. That's phenylketonuria. Okay, but all that she wants you to know here is that phenylalanine is converted into tyrosine by adding an OH group. See how the structures are basically the same? Here you just have an extra OH at the side. Entirety. Now we have threonine, serine, and glycine metabolism. We already said serine and glycine are interchangeable. So you can change it from one to the other. And additionally to that, threonine can be converted into glycine. Okay, and glycine is going to give us as a byproduct CO2 and ammonia. Again, we're just going to go to the Urea cycle, and then everything's going to be sorted from there. And you can also create oxalate. And I want you guys to keep this in mind because it's actually very interesting. Um, so oxalate is very important, as we'll see in a second, in the body. But very, very high levels of oxalate, once they're excreted in urine, what's going to happen is they're going to bind to calcium, and that's going to create kidney stones, which are very painful. And so because of that, here you can see the major reaction of glycine is degradation in human tissues, and it is one of the sources of carbon units for tetrahydrofolate. Okay, so tetrahydrofolate, again, we'll talk about in a second, but glycine is important for restoring the carbon units. Now, again, to the important part, kidney stones. So for those of you guys that are Friends fans, this is probably every time since first year that I've learned about kidney stones, this is the first thing that pops in my head. But calcium oxalate is one of the major components of kidney stones, and it's the most common type of kidney stone. And so like we said, glycine, when it's degraded, it's going to be converted into glyoxylate, and then this can be converted into oxalate, and then oxalate can bind calcium, and that's going to cause kidney stones, which is, very painful. And yeah, I just thought this was also an interesting clinical correlation. All right, on to the next. All good, everyone? Yes? Okay. Alhamdulillah. So now we move back to the thing that I told you that I touch upon again and keep it in the back of your mind, which is methionine and cysteine metabolism. So methionine, remember, is one of the essential amino acids. Private Tim Hall is the M. And methionine is exclusively glucogenic. Okay, and so it's degraded into succinyl-CoA, which is one of the cofactors in the citric acid cycle. Okay, and so why is it important? It's converted into something we call SAM, okay, or S-adenosyl methionine. Okay, and okay, why do we care about S-adenosyl methionine or SAM? SAM is a major methyl group donor, and it's a very high energy molecule, and it carries out a lot of important processes in the body. Okay, and when we say S-adenosyl methionine, it's because the S has a sulfur. Okay, and so we know methionine is now important for conversion into SAM, and it's also important for conversion into homocysteine. And we mentioned before, and I'll just reiterate that homocysteine is clinically significant because high levels of it can cause vascular disease, and it's inversely associated with vitamin levels, right? This is the exact same slide. I just added it again to re-emphasize that. Okay, and so let's go back to methionine and cysteine metabolism. So if you guys know this guy, Sam Burns, he died in 2014, but he had a very interesting disease called progeria, which is when you have a problem with P53. You guys don't have to know about this. I just, found it interesting. And it's basically their bodies age very fast. So his age, I think, might have been, a bit younger than 16, 17. No, I think before that, maybe 14, 15. But he looked very old because of this disease he had. And his name is Sam, so, just link it to remembering that Sam is a very important molecule in the body. And his TED Talk, if you guys do end up watching it, is very meaningful and very interesting. So like we said, back to our topic, methionine condenses with adenosine triphosphate, or ATP, and that forms SAM, which is a very high energy compound. So methionine with ATP is going to give us SAM, which is this, very powerful molecule. And the odd thing about it is that although it's very high energy, it doesn't have phosphate. It's not like ATP. You see how all these phosphates are excreted? And we add ATP to this molecule in order to generate SAM. So now we have, can I show you? Okay. So there's a methyl group attached to the sulfur molecule here. Okay, and once it's attached, SAM is considered activated. So methyl transferases, okay, can be used in order for SAM to be a beneficial molecule. It's a methyl donor, so it's donating these methyl groups, which is what makes it a very powerful molecule. So it's hydrolyzed, or it's broken down into homocysteine and adenosine. Adenosine can then be used to make adenosine triphosphate, monophosphate, our energy source, ATP, ADP, AMP, etc. However, if there's a deficiency of methionine, remember, which we said is essential in the diet, you can remethylate homocysteine to methionine. So it can feed back the cycle. And I know it's hard to visualize, but I'll show you a picture in a moment. Okay. However, if there's enough methionine in the body, the homocysteine is converted into cysteine, which we said, remember, methionine can be converted into cysteine eventually, and that's through conversion into homocysteine and then back down. And I'll show you a picture in a moment. Okay. So this is our methionine. We said by breaking down ATP, this can be converted into SAM. Okay, and SAM is going to donate methyl groups to methyl acceptors. Okay, and then as it keeps donating, eventually it's going to form our homocysteine. Okay, and now if there are low levels of homocysteine, of methionine in the blood, homocysteine is going to be converted back into methionine. However, again, keep this in mind, it's important. This process requires vitamin B12 as a cofactor. If vitamin B12 is not present, the whole thing can't happen. It's just kind of going to sit there and it's going to be useless. And remember, homocysteine levels building up are bad for your arteries. Okay? However, if there's enough methionine and everything is fine, then it can go to create cysteine as an amino acid. Okay? So again, just keep that in mind. Homocysteine can be converted back to methionine if levels are low, but this requires vitamin B12. No vitamin B12 means it's not going to happen. Ready? So again, this is just a figure to help visualize that for you. So we said we start with methionine, we take an ATP and it becomes SAM. Okay, it's going to keep converting, or it's going to keep sending methyl groups back until it becomes SAH, and then that becomes homocysteine. And what's the fate of homocysteine? If methionine levels are low, it can be converted back to it. However, we require vitamin B12 and vitamin B9, which is folate. Remember, folate is also what we said was important for spina bifida. Okay? However, if methionine levels are adequate, then it can be converted into cysteine. But now the problem with cysteine is that it needs vitamin B6. So do you see how it makes sense? If there are low levels of these vitamins, what's going to happen? You're just going to create a bunch of homocysteine, which is bad for your arteries and can accelerate atherosclerosis. So the whole process biochemically makes sense that supplementing with these vitamins would decrease those bad outcomes or the risk factors. So again, one more time, or okay, we'll go through the slide and then the other figure is also good. So cysteine is derived from methionine. We already said that. It's an essential amino acid. This is just repeating the old slides. Okay? And it can combine with serine to form cystathione, and then cystathione is hydrolyzed for alpha ketobutyrate and cysteine. Okay. So again, the key things here, which I'll show you guys here. So methionine can be converted into SAM by adding an ATP, right? And then it's going to donate its methyl groups, okay? And then it's going to form homocysteine. Now, homocysteine can create methionine, but it's going to need folate and B12. So vitamin B9, vitamin B12. Okay, and that's going to convert to methionine and tetrahydrofolate, and this can be used to, cycle back through this equation again. Homocysteine, if methionine levels are normal, can be converted into cysteine, but it requires vitamin B6. Okay, easy enough? Hope it makes sense, guys. Now, what is cysteine used for again? Just to complete the whole equation. To my knowledge, it's one of the glucogenic amino acids. And I'm not sure which processes exactly, but it's important for specific biochemical processes in the body. But this whole, equation, different vitamins that are needed, does this make sense to you guys? Or do you want me to repeat it one more time? All good? You sure? Double check. We're almost done. And this is just another clinical implication of the S-adenosylmethionine, which is SAM. As we said, it's a methyl group donor, right? And so it can be used to create epinephrine from norepinephrine, which is again, epinephrine, norepinephrine, these are our fight or flight hormones or our stress hormones that are very important, increase with cortisol and other things. But just again, this is one of the uses of SAM amongst a lot of other things. Okay, so again, cysteine is made from a carbon skeleton of serine and sulfur and methionine. So where is it here? Okay, so cysteine, we have our sulfur, and this is our serine group. Okay, and it's taking this sulfur from methionine. All right, and so cysteine catabolism is going to release an organic sulfate, which is excreted in the urine as sulfate. Again, I'm not sure how important this is for you guys, but it's mentioned in your lecture, so I thought I'd mention it here as well. And now we have our last two slides. So the metabolism of histidine. Remember again, histidine is one of the essential amino acids. It's the H in the mnemonic private Tim Hollis. So you have something called FL-glu or non-formamino glutamate. Okay, and the reason this is important is that it reacts with uh, tetrahydrofolate again. Remember, we talked about tetrahydrofolate and that it was formed from here, this reaction. Here is tetrahydrofolate is formed by the reaction of um, homocysteine turning into methionine, okay, with B12, vitamin B12 present. So, if folate, remember, because we said it's important, you need vitamin B9 and vitamin B12 for that conversion to happen. If you have a deficiency in folate, logically, this is going to build up, right? Because this reaction can't happen. It can't convert this from histidine all the way to glutamate, alpha ketoglutarate, and all of these things. So, okay, it's a deficiency. Why does that matter? Well, we have vitamin B9 deficiency and vitamin B12 deficiency. And one of the important things for us to differentiate clinically is understanding what vitamin this patient is deficient in. Because the way you treat with vitamin B12 is different than the way you treat with vitamin B9. But like you guys learned in HLS, both of them can cause megaloblastic or macrocytic anemia. So what we can do is we can use excretion of this molecule. So again, if there's an X here, if it's blocked, if there's no tetrahydrofolate, then this is going to increase. So we can test for excretion of this molecule because it makes sense. If there's a lot of it building up, a lot of it is going to be excreted. So we can test it to confirm that there is a deficiency. Okay, so as mentioned here, the N5-methyl THF, okay, is used in the remethylation of homocysteine to methionine, a reaction that also requires vitamin B12. All they're saying here is that this N-formino whatever is required here to form tetrahydrofolate and methionine. That's exactly what we already said. They're just mentioning it in words. Okay. However, this remethylation happens, okay, is the only time THF carries and donates a muscle group. So this is the only process of remethylation of this. So what they're trying to say is that these two are interchangeable. This turns into tetrahydrofolate, turns back into N5 for a minute tetrahydrofolate, and goes back. So they're kind of like a regenerating themselves. Okay, and why do we care about this molecule? It's used in the synthesis of purine rings. and you guys are going to learn about that in a later lecture. But the key thing that I want to make sure you know is that why do we have, let's say, a megaloblastic anemia, or why are the nuclei hypersegmented in those specific conditions? Because these molecules are very important for the synthesis of DNA, as mentioned here, synthesis of the purine rings. Okay, and so that's why folate and vitamin B12 can present as a megaloblastic anemia, because you don't have as much ability to synthesize DNA of DTMP and DUMP. Again, you guys are going to learn that a bit more later. But that's just the one thing that I want to hammer in for you guys. Histidine can be broken down eventually to form glutamate, tetrahydrofolate, or whatever. But this intermediate FL glue excretion test can be increased in folate deficiency. Why? Because if you don't have tetrahydrofolate, then you cannot proceed with this reaction. And we also know that tetrahydrofolate is important for this reaction to regenerate, because this is converted from homocysteine. This is homocysteine plus N-formynyl tetrahydrofolate gives you methionine and tetrahydrofolate. Again, I'm just reiterating the same reaction. So when you don't have this, when you don't have tetrahydrofolate, this excretion is going to increase. Also, you're not going to be able to form purine rings, which is why there's impaired synthesis of DNA and folate and vitamin B12 deficiencies, and that's why it presents as a megaloblastic anemia. I'm going to repeat that one more time because I felt like it was a bit confusing. So we know homocysteine can be converted back to methionine, but it requires vitamin B9 and vitamin B12, and it's going to give us tetrahydrofolate as an end product. This tetrahydrofolate can be used to convert a molecule from breakdown of histidine to form glutamate and alpha-ketoglutarate and reform this N-formynyl tetrahydrofolate. Why do we care about that? Because this is important to make purine rings, and this is important for us to create DNA and synthesize it adequately. For that reason, if there's a deficiency in tetrahydrofolate or folic acid, the levels of this can go up, which can help us diagnose that this is a folic acid problem. And secondly, that's going to lead to impaired synthesis of purine rings, which can present as megaloblastic anemia in both vitamin B12 and vitamin B9 deficiency. Does that clear everything up for everyone? Sure? There's one slide left in this whole lecture, because I just really want to make sure that you guys understood everything, and don't want me to repeat anything I will. Okay, so the last slide is the propionyl-CoA pathway. So in addition to other things such as valine, isoleucine, methionine, threonine are all converted into succinyl-CoA, and the way they do that is by this propionyl-CoA pathway. So this is propionyl-CoA. Eventually, it's converted into succinyl-CoA. Okay, and why do we care about this? Again, this requires vitamin B12 is a coenzyme. So if there isn't vitamin B12, this process is not going to be able to take place. Okay. And yeah. So this is, again, succinyl-CoA is an intermediate in the citric acid cycle. So I know there's a lot of complicated processes in these lectures, but I just hope you see that at the end of the day, everything feeds into something else. You know, that's why they're considered biochemical pathways. Everything is continuous, subhanAllah, in the body, and if you have one vitamin deficiency, it's going to affect these processes, which is why you get, you know, the clinical presentations and the symptoms you see, whether it's phenylketonuria, increased levels of homocysteine in vascular disease, vitamin B, megaloblastic anemia. So that's, you know, the clinical takeaway from all of these lectures. So again, just to recap, vitamin B6 is important for various reactions, as well as vitamin B12. So we said vitamin B6 is important for converting homocysteine into cysteine. Okay, and we have vitamin B9 and vitamin B12 that are important for conversion of homocysteine and back into methionine. And also a deficiency in those cause megaloblastic anemia. Also for vitamin B12 specifically, it's deficient. Its deficiency will lead to impairment and formation of succinyl CoA, and you can also have folate deficiencies associated with spina bifida. I think there's one more that I might be missing if you guys remember. But I know the vitamin parts can be confusing, so I would suggest just write them down on a sticky note, keep them in your notes so you can always distinguish this virus versus. Do you guys have any questions? Yes, no? And again, don't forget to know which are the essential amino acids, ketogenic, glucogenic. You guys could be asked about that. If I find any additional questions for this lecture, أكيد I'll find صراحة. I had an exam yesterday, so I didn't have enough time to do all of it. But inshallah, if I do find, I will share with you guys. And if you guys have any questions, send me an email. My email is in the last PowerPoint. And yeah, يعطيكم العافية. Thank you for attending. I'm sorry if it was a bit of a longer PAL. But inshallah, ya Rab, it helped you guys out. That's what's most important. الله يعافيكم. Have a wonderful rest of your day and enjoy the rest of the PALs. And again, I'll try to add some more questions and I'll share the PowerPoint with you guys. Okay, I think we can end the recording on that note.